Let's do Kahoot.
Perfect.
Okay, wait. Wait, how do I do this? Play. Never done a Kahoot before. Hold on. I do classic, right? Yes.
Yeah.
Okay, there we go.
Awesome. Has anyone not played Kahoot ever before? Assume it's standard, but maybe not.
Good question. When you see the screen, is it like, do you see the boxes like on the side of like speakers?
No, it's
Are
just
they
it's just the Kahoot game.
Okay, perfect.
And I can monitor chat too.
Thanks. Brody, stop. What? Are you kidding? Okay, that's enough out of you. Nope.
We lost dog privileges.
No, he ripped apart my polar bear that I got at the San Diego Zoo. He just ripped the nose off. Gosh. Don't eat my charger. Stop.
It's this toy now.
Well, now it's destroyed, so. Is that, okay, wait, we'll wait a couple minutes because it seems like people are still joining. I brought his own toy with me in here because I knew he wanted to come, and he still decided that my stuffed animals were better, so.
Well, obviously they're not his, so they must be better.
Brody. Hey. You guys hear me yelling at my dog throughout this? I'm so sorry.
Is anyone like struggling to join? Should be like 3 more people, unless people are just watching.
That's fine. Should I start then?
Yeah, that's fine.
Sure.
Let's get going. All right, let's do this.
Go. Disulfide bonds are present in which protein structure?
All right, so after questions, ideally we're going to try to get you guys to explain why something's true or false, or like the correct answer. There'll be a lot of true and false questions, I think.
Yeah, there's a good amount.
But yeah, I mean, if something's like easy, we'll explain it, but we want you guys to explain the concept as much as possible.
Is that everybody? Oh, there we go. Tertiary and quaternary structures. Yeah, good job.
Good job.
Does anyone know why it is not? Well, it's not secondary structure because disulfide bonds are only present between, I don't want to say it's wrong, domain, no. Disulfide bonds are present in both tertiary and quaternary structures because in secondary structure, we're dealing mainly with hydrogen bonds between the backbone, and primary structure is covalent bonds between amino acids.
Mm-hmm.
Sorry. Okay. Does anyone have any questions on this one? This one's kind of basic stuff. Just got the next one. Nina's in the lead. All right. Which level of protein structure would urea, which disrupts hydrogen bonding, have the largest effect on? Primary, secondary, tertiary, or quaternary? Good job. Secondary. Yeah. Go ahead, Jason.
Yeah, good job. Which other structures could it also have some effect on? I said largest, but which other structures could it have some effect on? Anyone want to answer that?
Could it also affect the tertiary structures?
Yep.
And in some proteins, it would affect quaternary as well, because not all proteins have quaternary structures, but it could, if they do, it could affect that as well. Good job, guys. Ooh. This is a multi-select. Which of the following may be considered an example of tertiary protein structure?
I didn't even know you could do that in Kahoot. Oh,
I know.
You're leveling up.
A fun new feature I found. Nope. Not allowed in my room. He is, but he's just being fresh.
How old is he now?
7 months, I think.
Nice.
I know, he's a baby. I think we're waiting on one person to answer. Is there a way to like, no, it'll skip the question, I think.
I think you can hit skip.
Okay. So we have 8, red and yellow, which is correct. Tertiary protein structure. So interactions between two R groups located far apart on a polypeptide and disulfide bonds between cysteine residues located far apart. Can someone tell me why it is not hydrogen bonds between backbone amino and carboxyl groups? Put it in the chat, or you can say, or we can talk about it. I guess I'll just talk about it. So hydrogen bonds between backbone amino and carboxyl groups is more related to secondary structure. Tertiary structure can be anything between the two R groups located far apart on a polypeptides, a kind of a key thing because it's in both of them, but it is something that like with tertiary structure, because you're dealing with the whole protein or the whole polypeptide, they can be far apart, but that's why red and yellow are the correct answers. And if you have any questions, like feel free to stop us at all also. Ooh. Okay. True or false? RNA polymerase can bind to DNA on its own.
Good.
Good.
It's false. What does RNA polymerase need to bind to DNA? Chat or say it.
I mean, isn't it just like transcription factors?
Yep,
Mhm.
exactly.
Good job.
What are the transcription factors used for RNA polymerase in eukaryotes? I know in the prokaryotes, it's usually like Sigma 70. Is that the same for eukaryotes or is it something else?
It is something else. It's called the basal transcription factor.
Oh, right.
Sigma is only for prokaryotes.
Yeah.
All right, another true or false. The backbone of a protein would be broken when placed in a solution with a pH of 2.5.
Is there a way to find the code? Someone just joined.
It should be, hold on, let me go exit full screen. It's at the bottom, the 1822580.
Yeah, I'll put it in the chat.
Okay. So this is pretty split. The reason it is false is because this is kind of a trick question. When you first read it, it's like, oh, I don't know about pH at 2.5, but that's kind of irrelevant in this question because the backbone, if you remember, the backbone of the protein is the primary structure, and primary structure is not altered by environmental changes such as temperature, pHs, or like solutes or whatever. The backbone of the protein would not be broken because the pH will not affect the backbone because it's primary structure. If this was like, would secondary structure be altered? Maybe, but you need more information because that's a pretty acidic pH, and some proteins would be able to like not unfold in that. But in this case, because it's asking about your primary structure, the pH will not affect that. Anyone have any questions on that?
Yep. The key thing here is covalent bonds. So the backbone is held by covalent bonds, and it takes in this class it takes a ton to break a covalent bond. So it wouldn't be broken. But like something like tertiary or quaternary would be affected.
Right. Ooh. I wish we could like offer like extra credit on your exam for like people who like win, but we can't. We don't have that power. I feel like it would have been kind of stressful too, but no. All right, true or false? Gaps exchange GDP for GTP in the cytoplasm.
Pay attention to where these are happening too.
Jason, are you typing something?
I'm just fiddling with my keyboard.
Oh.
All right, I think we're good.
So yeah, this is false. What do gaps do? Anybody?
They exchanged GTP for GDP
Mm-hmm.
in
Yep.
the cytoplasm.
Yep. So location's correct here, but gaps are going from GTP, hydrolyzing it to GDP. So one thing to remember is gaps they don't exchange GTP for GDP. They hydrolyze GTP. So that means that that GTP molecule is losing a phosphate group. Whereas when we're going from GDP to GTP, we're diffusing a whole different molecule into that GTP is not the same as GDP. But regardless, gaps do not exchange GDP for GTP. They hydrolyze GTP to GDP.
Yep, I think it's key to look at the names here. So stands for GTPase activating protein. So it activates the intrinsic like GTPase activity of RAN. So that means it allows RAN to hydrolyze GDP to GDP. In contrast, GAF stands for guanine nucleotide exchange factor. So it exchanges GDP for GTP, and that happens in the nucleus. Anyone have any questions about that?
Ooh, Christine is a streak. All right. Which of the protein models would you use to show secondary structure? Backbone, ribbon, ball and stick, space filling. These are the type of questions that that she'll expect you to know on the exam for anything with protein models. Like all you really need to know is like what they show and like what or what they're supposed to show or like what they can tell you. She's never really going to ask you too much detail about them though.
Mm-hmm. Yes, it's more likely like they'll be used in a question, but you won't be asked a question specifically about it.
Right, right.
And I forgot to do it at the beginning, but at the end we can talk about like strategies for the exam and what it's gonna look like too.
Oh yeah, we could do that too. We talked about that like in between also.
All right, I think we're good.
And if I skip it, will it? Okay, wait. Let's see. Okay, there we go. Yeah, ribbon's the correct answer. There's no really like explanation. Like, there's no way. Like, you just kind of got to memorize these. I don't have like unless you have anything, but like
Yeah,
about memorizing. But
I don't. But
yeah,
also,
these are basically just memorization. But she's really like, she'll only ask it like briefly in a question because remember your exam's all open response, so it's not going to be something like this with like multiple choice questions. But she could mention it like within your the question, and you might have to like elaborate on it. That's pretty much it. Haley's making a comeback. All right, true or false? The R groups of the amino acids in the alpha helix are available to make interactions with other molecules. Music's so dramatic. I don't know if you guys can hear it, but good job.
I missed the
Woohoo.
OG Kahoot music.
I know, me too. Yeah, good job. This is true. Yeah, they stick out. They're available.
Cool.
Woohoo. All right. Another true or false. Proteins start translating when ribosomes are docked to the ER membrane. What?
What was music?
I know.
Pop.
Yeah, this is
Nice.
false.
Good job, everyone. So, basically, they like the mRNA gets to the ribosome in the cytoplasm, and then something, I forget what it's called, something, signal recognition particle, I think, SRP,
Yeah.
drags it to the ER membrane where it can then be translated there. If it has an ER localization sequence, it'll go into the membrane. If not, it'll go into the cytoplasm.
Haley's in the lead. You get a hypothetical extra credit. If you mutate your protein of interest at the 21st amino acid, is this amino acid closest to the N or the C terminus?
Rachel, do we still have like biosci t-shirts?
We do. I could give you a biosci t-shirt.
Or like a, yeah. I have like UPC socks if anyone wants.
We get a virtual high five. You win. All right, yeah. So this one's kind of tricky. It is the
What's
N
up,
terminus. Because anytime you get a protein, you should, unless specified, you should assume that your protein is at least 100 amino acids long or more, because usually proteins are a lot more than that. They're usually around 700 or how many ever amino acids long. Unless she specifically is like, your protein is 30 amino acids long, then your answer would be the C terminus. But because there's no specification, you should assume that the protein is at least 100 or more amino acids. So 21 is closer to the N terminus in this case. Hope that makes sense. Oops, so sorry. True or false? A protein needs both ER localization sequence and the ER attention sequence to be secreted outside of the cell. Yeah, this is false. If it has an ER retention sequence, it won't leave the ER.
Mhm.
If it had an ER localization sequence and you weren't giving any other information, it probably would be secreted outside of the cell because it would go through that endomembrane system. But because it has both, it will go into the ER and it will stay there.
Any questions on that? It can be a little tricky.
Yeah, definitely. I don't know if you guys have seen that chart. Remember the flow chart, Jason? It's in
Yeah.
the slides we are going to send out.
Yeah, I'm gonna send a. It's a great chart. I would like any kind
Really
of type of question
helpful.
like that. Just like follow that chart, and you'll get to
Yeah.
the right answer.
Yeah, no, literally, the chart is so helpful.
I'll send out that slide after this.
Mhm. Protein with one polypeptide can contain multiple domains. Is this true or false?
Domains and subunits. Yay.
Woohoo!
True. Proteins contain lots of domains, like especially if it has like multiple functions. One could be like to open an ion channel, another could be to activate something else. So proteins contain as many domains as they can fit, pretty much.
Protein with one polypeptide cannot have multiple subunits.
Because that is a subunit.
That is one singular subunit. That is the difference, but it can have multiple domains. I know subunits and domains can be kind of tricky. Woo! All right. What charge are acidic amino acids at physiological pH, which is 7.4? Acidic amino acids. Ooh. All right, so yeah, this is negative. Acidic amino acids at physiological pH are negative. They're not neutral, and they're not positive, they are negative. Your basic amino
Oh.
acid, what? No, go ahead.
I'm just gonna say, don't worry too much if you didn't get this. I don't think Laura would ask like something like that. She would be like,
No, but she could ask like if you have like a like more like if you have an amino acid with R groups pointing outward and like a negatively or positively charged like area, it would be mostly like acidic because they're negative and they're being attracted to opposite charges. She wouldn't ask you specifically like this question, like what charge are acidic amino acids at a physiological pH, but it could be something like brought up within a question. So it's just easy to, it's just a nice thing to remember that acidic amino acids are negative at physiological pH, basic amino acids are positive, and everything else is neutral. So all your non-polar amino acids are neutral. So there's three sections of polar amino acids. You have your polar uncharged amino acids, your acidic amino acids, and your basic amino acids. Those are all polar. It would specify that if it's a polar uncharged, but she might, I don't know if she's going to ask you more because you have access to like the amino acid charts, but she has in the past. So, but yeah, so it's just an easy thing to quickly just jot down or remember when you're taking your test in case it comes up. But yeah, she won't ask you something specific like this, but it wouldn't hurt to know.
I would say most of the questions are going to be like about interactions with types of amino acids, like how positive charged amino acid would interact with other like proteins or amino acids, whatever.
All right, true or false? Introns are transcribed but not translated. Woohoo! Yeah, that's true. Introns are transcribed, and then through that post transcriptional or RNA processing, it gets cut out during splicing. Introns do not code for any genes. They're kind of just like dead space. So those get cut out after transcription and before translation. So they are transcribed, but not translated. Exons, however, are transcribed and translated because those are not cut out by the spliceosome. Good job. Woohoo. All right. Some multi-select. Which of the following are ways that the cell responds to unfolded / misfolded proteins?
Favorite type of question.
Yeah. So I gave a good amount of time to think about this one.
Just think about like what if there's unfolded or misfolded protein, what does the cell want to do to fix that?
We want to upregulate translation and make more proteins. Do we want more chaperones? Do we want more proteins into the ER? Do we want more lipid biosynthesis? Remember, lipid biosynthesis is something to do with the amount of space available. The ball is bouncing at the beat of this music. Just watching it. Give another couple seconds and then we'll go. Jason, have you seen the TikToks where they use this song?
No, what are they doing though?
Oh my God, it's so funny. It's so funny. It's like the original Kahoot music. All right. All right.
At me on TikTok.
Oh my God, I need to. Yeah, you're right. All right. So let's talk about this one. So we increase chaperone production and we increase lipid biosynthesis. These are two things we want when the cell is responding to unfolded or misfolded proteins, because increasing chaperone production, what do chaperones do? They help fold proteins correctly or like help misfolded proteins fold correctly. We want to increase lipid biosynthesis because that increases the space available within the ER to work with these proteins and to fix them. We will not want to upregulate translation because we don't want more proteins being made when we're trying to fix the ones that are already unfolded or misfolded. So we don't want more added to that. So we wouldn't want like more coming in, and we want to focus on what's already unfolded and misfolded before we make more proteins. So we would want to actually down regulate translation. And then we don't want to translocate proteins into the ER because that's where we're doing this unfolded protein response. So we want to translocate proteins out of the ER. Does anybody have any questions on this one? No. Okay. All right. Trip genes are blank in the presence of tryptophan, on or off?
We love our trip opera on question.
Yeah, they're off. Remember that we don't want to make more tryptophan if we already have enough of it. So the trip genes will be off in the presence of tryptophan. They'll be on when we don't have any tryptophan. Ooh. Which of the following is affinity not dependent on? Number of bonds, strength of bonds, angle of bonds, or KD. Affinity does not depend on one of these. Can't believe he ripped apart my stuffed animals. It's KD.
Yeah. So for this one, KD is related to affinity. Like affinity is kind of like, or KD is kind of like a measure of, it's not directly a measure of affinity, but it's like related to affinity, but affinity doesn't depend on KD. Like it's just a measurement.
It does, however, depend on the number of bonds between the protein and the ligand and the strength of the bonds. I actually don't know if do you know if it depends on the angle?
It does.
It does. Okay. Yeah. So those, it does depend on all of them, just not KD. KD is that thing where it's like if you have a higher KD, it's lower affinity, or lower KD, higher affinity. So they have a relationship, but affinity doesn't depend on KD.
Any questions on that? Or KD in general? I know it's a little tricky.
She doesn't spend too much time on enzyme stuff, but she could ask you a question about it. I know there was a problem set question about it. So anything on the problem sets or on the practice quizzes are fair game for her to question you on. Attenuation will occur when only when trip levels are high. Is true or false? Yeah, this is false. This is kind of a trick question. So attenuation, so say we start with high trip levels, which is what this question is referring to, because it didn't specify that this is after we've started transcription. If trip levels are high just at the beginning, transcription will never occur and attenuation won't occur, because attenuation will only occur once transcription has begun for the trip operon. And then as we're transcribing, levels reach this threshold where it now becomes too high. And that's why attenuation will occur, because now we have enough tryptophan after we've started transcribing. And then attenuation will occur to stop it. So basically attenuation occurs when we start with low levels of tryptophan. And then as we're transcribing, those levels get higher and higher and higher until we no longer need any more. And then attenuation will occur to stop transcription. But if we're starting with high levels of trip, which is what this question is referring to, attenuation won't occur because transcription will have never started. Does anybody have any questions on this? Because I know the trip operon can be tricky. I know that one's kind of a trick question, but it's mostly to help you like think about different conditions and when what is what. Which of the following is not true about enzymes? They increase the rate of reaction by decreasing activation energy. They make and break covalent bonds. All enzymes are 100% protein, and they decrease the rate of reaction by lowering activation energy. Oh my God, the music's so dramatic. Yeah,
Good job.
well,
Sorry. I read that wrong. So
what is not true?
sorry, trick question.
One person who did get it right, that is the correct answer. They do not increase the rate of reaction by decreasing activation energy. Wait, did we write this wrong?
Oh,
I thought they increase the rate of the reaction by decreasing the rate of the activation energy, because that makes it go faster.
Yeah, they do. I
Did I do that
selected the wrong one as the correct answer.
okay.
Yeah, I'm pretty sure it's supposed to be the green one.
It's green. Yeah, you're right. I was like, wait, that's not right. I clicked the wrong one. Sorry, guys.
I need my points, please.
Sorry about your streaks.
Yeah, so sorry about that. I'm sorry that you won't get a virtual high five. Yeah, no, it is supposed to be green. I think what happened was I wrote them in backwards, so I clicked the wrong one as the correct answer. I'm sorry. You guys are right. They do decrease the rate of reaction. They do not decrease. Okay, they do not decrease the rate of reaction by lowering the activation energy. They increase the rate of it. So that, wait, yeah, yep, it's green. I'm sorry. Next. Okay, repressible operons are usually on and are turned off by the presence of a specific molecule.
What is trip operon?
True, this is trip operon. See, I understand the trip operon, but I like still to this day, even after taking the MCAT, do not understand the lac operon. And I remember learning it in 151. And
I remember learning about it. Isn't it like just like backwards of trip operon or something?
It's I don't like it. I do not like it.
I don't remember it. I think I only talked about it in like genetics.
Yeah, I had to relearn it for the MCAT and it was awful. Yeah, repressible operons are usually on and are turned off by the presence of a specific molecule. This is the trip operon. It's usually on and then turned off by the presence of tryptophan. The opposite of this is the lac operon, which is an inducible operon. Inducible operons, which you don't need to know, but just because she won't test you on this, but inducible operons are usually off and are turned on by the presence of a specific molecule. For the lac operon, that's lactose. But the trip operon is repressible, so it's always on unless there's tryptophan, and then it will be turned off. Okay. Which of the following is not an example of post-translational modification?
Hopefully you did this one right.
I hope I did too. I'm kind of nervous. Guys, don't tell Laura. Okay, I did it right. Yeah. Methylation, acetylation, and ubiquitination are all examples of post-translational modification. Transcriptional repressors happen during transcription, so that wouldn't be after translation because transcription comes first.
Before translation.
Yeah. Woohoo. All right. RAN is a GTPase. True or false? RAS, RAN. Do they know RAS yet, or is that the next unit?
Yeah, I got a bunch of questions about like what RAS is. RAS is in the same family as RAN.
Yeah.
We'll talk about that soon when we get to like cell division, but it's the same kind of enzyme.
Yeah, that's the next unit, I'm pretty sure.
And also pay attention to what RAN does because it might come back in future questions. People got mad last semester because they didn't remember it came up
Oh, yeah, they did.
in the first exam. So remember what guess and gaps do for, it might come up later too.
Yeah, RAN is a GTP ace. What is a GTP ace? What does that do?
Someone answer.
Answer and you get your points back. I'm just kidding. I have no control over that. Anybody? No one knows what a GTP ace is? Nice.
Is it an enzyme that changes GDP to GTP?
Mm-hmm. Yep, it hydrolyzes GTP to GDP. And ATPase is the same thing, but with ATP. So RAN is a GTPase. It does have that intrinsic ability to hydrolyze GTP to GDP. It just does it very, very slow, which is why when we talk about nuclear import and export, it needs that gap to help increase the rates of it hydrolyzing GTP. Good job. All right, true or false? The mitochondrial signal sequence is cleaved off after transport and the ER signal sequence is not.
This was in one of her like extra videos that she posted. So definitely check that one out. It's actually super useful.
Yeah, definitely. Jason, I forgot about them getting mad last semester on the final with the RAN question.
A bunch of people were like, "What is Geth and Gap?" I was like, "We talked about this already." They were like,
Wait.
"We've never mentioned this before."
No, I know. I didn't write this one wrong, did I? No, I did not.
Yeah, that's true. Mitochondrial signal sequence gets cleaved off as soon as the protein enters the mitochondria. That it talks about it in the video I was just talking about. And ER signal sequences do not get cleaved off for the most part.
Yeah. All right. Which of the following properties of a protein is least likely to be affected by pH? Huh, is this something we talked about already? Kahoot just reminds me of high school Spanish class.
Exactly.
Like
That's what I keep thinking about. It's like
They're
where I
like,
played it the most.
oh, yeah. For anyone who was wondering, not that any of you were, Jason and I did go to the same high school. So yeah, this is primary structure. Primary structure is least likely to be affected by environmental factors such as pH and temperature, UV light, all that stuff. Because for the purposes of this class, covalent bonds are the strongest bonds that we talk about, and none of the environmental factors will affect it. But it could affect secondary, tertiary, and quaternary structure because it could unfold the protein. Good job. Ooh, okay. Where does the repressor bind in the trip operon? The repressor is what stops it. Promoter, operator, attenuator. Does it bind to the RNA polymerase?
Key thing here. Well, not for this question, but the promoter for the trip operon is different than the promoter for the trip repressor. There was a question about that last semester, and a lot of people got that mixed up. So like the trip repressor is separate from the trip operon, and they have their own promoters.
Yeah. But yeah, the repressor binds to the operator. The RNA polymerase binds the promoter. Nothing binds the attenuator. That's not related, and nothing binds the RNA polymerase. But yeah, the repressor binds the operator to stop transcription if there's enough tryptophan around in the cell. It's pretty stagnant. All right. Okay. Two different proteins have the same domain in their structure. Which of the following is true? Jason, I forgot, did you get your COVID vaccine?
Yep, I got my second dose.
Woo, yay. Is that for Boston?
You're the Moderna, right?
Yeah, what'd you get?
I had the Pfizer and I had no symptoms like at all.
I was like, okay. I just was like achy and stuff. But I got like better throughout the day. Like I woke up feeling like really nauseous, but then as the day went on, I was like perfectly fine by like dinner time.
Mm.
It was weird.
I just had like the normal sore arm. It wasn't bad.
My arm hurt more in the first one than it did the second one, which is weird.
Yeah, same for me.
All right. Yeah.
So for this one, the key like detail is that domains are evolutionarily conserved. So that means that the domain in like, I don't know, like a frog or something, maybe for the same protein might be similar to a domain in a human. So that means that yeah, they'll have the same like part of the same structure.
True or false? A protein can enter the nucleus while fully folded. Yeah, this is true. Remember, the nucleus has those nuclear pores. They're pretty big. So protein can't enter the nucleus while fully folded. So just remember the nucleus has those pores. So it doesn't have to unfold. Okay. Is the trip repressor protein a negative or positive regulatory transcription factor? Yeah, it's negative. Good job, guys. Everyone got that. Awesome. Misfolded protein with a nuclear localization sequence may not be able to go into the nucleus.
And we're gonna ask you why, so someone better be able to explain this one.
That sounded like a threat.
Yeah, it is.
You better be able to explain this one. Oh my gosh. True.
Nice. Let's talk about this one. Can someone explain why that might be true?
Jason's gonna beat you up if you don't. I'm kidding. For legal reasons, that's a joke. We're recording.
This is a guess, but like just because of the fact that it's misfolded, the cell is not going to want it to go into the nucleus, so they're going to want to wait until it's like fixed.
I mean, sort of, but what could happen? So if the NLS is like on part of the protein, what could happen if the protein is misfolded? Anyone could answer this.
The NLS might be broken.
Yep, exactly. It could be broken or it could be hidden. So it could be like on the inside of the protein when it should be on the outside. So it may not be able to go into the nucleus.
But yeah, that was a definitely good thought, like to approach it, because you knew something was about the chemistry of the protein would change with the misfolding, like not the chemistry, but like the way it reads the nuclear localization sequence. But yeah, basically.
This is the kind of question that Laura would ask. She'd be like,
Yes.
she'd make a mutation and then ask like what the effective mutation would be. So be prepared for questions like this.
Yeah, they're definitely make you think. True or false? A protein with an NLS can move back and forth between nucleus and cytosol. This is another very Laura structured question. Yeah, this is false. Good job. Remember, to leave the nucleus, you need an NES. And since there's no specification of an NES, it just says an NLS. The protein, we know it can move in, but we don't know that it can move back out. So this would be false. All right, true or false? Secondary structure is caused by hydrogen bonding between R groups. Think about this one before you answer. Yep. This is false because the secondary structure makes hydrogen bonds between what? Not R groups. Between.
Someone say it.
The backbone.
Yeah, the
Yeah,
backbone.
there you go.
Good job. True or false? Drosha turns pre-mRNA into, wait, PRI-mRNA into pre-mRNA in the cytoplasm.
About where this is happening.
I'm not the expert on this stuff. Jason is, so. Jason, this is you.
Drosha acts in the nucleus, not the cytoplasm. That's the key part of this question. It's hard to explain it with that diagram, but that's why it's false, because it happens in the nucleus.
True or false? A protein that enters the ER won't go back into the cytosol. Yes, it's true.
Yep.
Why do people say false? Just curious. It's not process for us. You don't want to answer, that's totally fine. But yeah, once the protein enters the ER, the odds it leaves it to go back into the cell, unless it's going to go through the endomembrane system and exit the cell, is like slim to none. The protein usually, once it's in the ER, does not come back out. Which is why this is true. Jason, do you want to add anything to that?
Nope, that's pretty much it. Once it enters the ER, it's kind of stuck there, or stuck in the endomembrane system.
Mm-hmm.
Wait, I had a question.
Wait, I'm so sorry. I do not know how to pause this. Well,
That's fine.
well, ask after this question. I'm so sorry.
Right.
You can ask now why people are doing this one.
Yeah.
So for like that last question, is the ER just like it's when she's, for example, she says the ER, are we assuming the entire endomembrane system or just the ER? Because I put false because I'm thinking it could go to like Golgi or like it could move, you know what I mean?
Right.
Like it could.
Well, so
The key part of that question was that it said back into the cytoplasm, because the cytoplasm is not a part of the endomembrane system. It will never, once it enters the ER and the endomembrane system, it will not re-enter the cytoplasm. If that makes sense.
So like the ER is part of the endomembrane system, so it will stay either like in the ER or somewhere in the endomembrane system. So it could go to the Golgi or somewhere else, but it'll still be in the endomembrane system.
But it will never be in the cytoplasm.
Yep.
I think so.
Yep, no problem. I thought it needed a retention sequence to stay in the endomembrane system. It needs a retention sequence to stay in the ER specifically. The endomembrane system and the ER are not synonymous. The ER is a part of the endomembrane system, but it is not the whole thing. So if you have an ER retention sequence, it'll stay in the ER, but if you don't, it'll go through the endomembrane system, which is like the ER, the Golgi, the lysosomes. It could exit the cell.
Yeah, if it doesn't have a retention sequence, it'll probably exit the cell, but it won't go back to the cytoplasm.
Yeah, once it leaves the cytoplasm and enters the endomembrane system, it will never go back into the cytoplasm. All right, so this one. A protein with an ER localization sequence and a mitochondrial sequence will end up where? It will end up in the ER because the ER localization. Okay, so for this one, think about it like as a protein is being translated, the part with the ER localization sequence comes out of the ribosome first. So that is read first. It's read like in order. So because it's read before the mitochondrial sequence, it'll just automatically go to the ER because it didn't even read the mitochondrial sequence because it comes after the ER if a protein has both.
By end up in this question, you guys didn't get tricked up by it, but end up I mean like go to like
Yeah.
if it doesn't have any other sequences, it'll probably be secreted, but it'll go to the ER first. It don't ever get to the mitochondria.
Mhm. All right. A protein enters the mitochondria fully folded. True or false? Yeah, it's false. Nucleus, it does. Mitochondria, it has to unfold and then it kind of gets let in like beads on a string, and then it will refold once it's inside. Good job. All right. What charge are nonpolar amino acids at physiological pH? This is another one of those. Just something that is helpful to know. These are MCAT questions. Yeah, nonpolar amino acids never have a charge. They're neutral. Acidic amino acids are negative. Basic amino acids are positive. Neutral, nonpolar. Boom. Good job. Okay. True or false? Every time a protein is localized to the endomembrane system, it must have an ER signal sequence. Yeah, good job, guys.
Nice.
So go ahead, Jason. No,
yeah, anytime it goes to the ER, it has to have the ER signal sequence. Otherwise, it'll. I mean, if it has like, if it doesn't have an ER signal sequence, it could go somewhere else. But anytime it enters the endomembrane system, it has to start in ER within the ER signal sequence.
It's I mean, the flow chart that we'll send out is like super, super helpful. But if say it's like the protein needs to end up in the Golgi, it has to go through the ER first. So it would still need an ER signal sequence. That's why anywhere in the endomembrane system, it needs at least the ER localization sequence because it needs to start there. But most of you guys got that right. So that was good. All right. A protein can leave the cytosol even if it doesn't have an ER signal sequence. Cytosol, cytoplasm, same thing. I used to think cytosol and cytoplasm were not the same.
Me too.
For the longest time, I literally was like, what is a cytosol? I did not know. And then now, whatever. Okay. Yeah, this is false. Wait.
Wouldn't this be true? Because can it go to
Did I do it wrong again? No, I thought it was false.
Can it go to the nucleus or like the mitochondria, even if it doesn't have an ER signal sequence? Does that count as like leaving the cytosol?
Okay, this was one of your questions, Jason.
I know, but
I think it was like to leave, I think it was to leave the cell, even if it doesn't have an ER signal sequence, because it can't leave the cell. A protein can't leave the cell if it, I assume this just meant the endomembrane system. Protein, wait. Yeah, a protein, I don't know. What were you meaning by this question?
I don't remember.
I think this is one of Laura's questions. Like, didn't you give us a list?
Yeah, I took some questions from like old exam reviews, so this might not have been like my question, but
so it might have been, it could have been mine. Okay, wait. A protein can leave. Yeah, it could to go to the mitochondria or the nucleus, but I think it's just not specified here. It would have to be in an exam, so you're never gonna get a question where it's like not specified where like this. But I think the meaning behind it was like about the endomembrane system, like to go into the endomembrane system, which is why I think most of you got it right, because you were thinking of the endomembrane system.
Right. That's probably like the context that the question was in.
I think
talking
it was in
about
another
the system.
context, yeah,
Yeah.
but technically, this is true in terms of like leaving to go to the mitochondria or the nucleus. So if you said true and that was your thought, you are correct also. But this is false in terms of the endomembrane system. So I think what happened was in our exam reviews before, we were specifically talking about the endomembrane system, and then we went on to the nucleus and mitochondria. So this is before that. So this would be false in terms of the endomembrane system. It can't leave the cytoplasm to enter the endomembrane system if it does not have an ER signal sequence. But if we're talking about like the mitochondria and nucleus, it could if it has a separate sequence for those specifically. Does that make sense? I'm sorry, this one was kind of confusing, but I'm realizing now that it was in another context when we wrote it. Yeah, okay, cool. So sorry, guys. I'm glad nothing's really at stake here. Otherwise, I'd give you all extra credit if I could. Okay, which of the following is not true regarding gene expression in prokaryotes? Prokaryotes regulate mRNA localization, regulate mRNA degradation, DNA content. Good job. Prokaryotes don't package their DNA. They just don't do it. They do regulate mRNA localization, they do regulate mRNA degradation, and they do regulate rate of translation, but they don't package their RNA, their DNA into chromatin. In the slides that we'll send out after this, we have like a nice chart of everything, like comparing between the eukaryotes and prokaryotes. That should be pretty helpful. But yeah, they don't package their DNA into chromatin. Haley has a streak. Woo! All right, true or false? GAFs exchange GDP for GTP in the cytoplasm. The music is sending me, I can't. Yeah, this is false. They do exchange GDP for GTP, but they do it in the nucleus, not the cytoplasm.
Yeah, pay attention to where things are happening. That was a key part of a question on the exam last semester. So,
Oh yeah.
and I graded that one, so I know a lot of people got that mixed up.
Yeah, it's just advice for her exams is just read carefully. You have what, 20? Does she do 36 or 24 hours?
I think
She's
it's
doing
36 this time, but
yeah, because she's doing 36
hours.
for 151 too. So I always get them confused because I'm TAing two classes. But yeah, so you have 36 hours to do it. I'm not saying take all 36 hours, but take your time reading the questions, think carefully about what she's asking you before you like immediately knee jerk and write a response, because you might miss something. But that's just the best advice I can offer for her questions.
If you're confused about the wording of a question, definitely email her. Or
Oh yeah,
she's also doing like help section or like question sections for this exam. So you can go to that and ask her like what she meant by a question.
mm-hmm. Because she's literally like last semester and for the winter semester, she was like on her laptop for like the majority of the hours that you have to work on it. So she like would answer emails for students who had questions like immediately, because she's trying to like simulate like an actual in-person exam. Like you could just go up and ask her. So she's trying to like be there to like answer your question. So if you definitely have a question, like don't even hesitate, like email her about it because she will answer. Why are R groups of the amino acids available to make interactions with other parts of the protein? Okay, yeah, we want to answer that question in a second.
Lucas, your question, yes. So if the answer to that question was ER, because the ER signal sequence is like ahead of the mitochondrial signal sequence on the DNA, so it kind of gets read first, like, and it gets co-translated into the ribosome, so it'll never get a chance to go to the mitochondria.
Give you 10 more seconds and then I'll skip it. R groups stick out. They're not tucked away on the inside. It's not none of the above. They stick out away from the helix. So that's why they're available to make interactions. Sometimes she really likes to ask questions about like interactions between R groups, like electrostatic stuff. She asked a few questions about that before. She usually like gives a diagram. So just know that they're sticking out and away from the helix. Just know how they're like oriented. But good job. When regions 3 and 4 form a hairpin loop, transcription of the trip operon will terminate. Yeah, this is true. Remember that this is attenuation. This terminates the transcription because we have enough tryptophan. When regions 2 and 3 form a hairpin loop, 3 and 4 are not available to form one together. So when 2 and 3 form a hairpin loop, transcription will most likely continue. So when 3 and 4 are together, that's why it will terminate. Love the trip operon, just kidding. Alternative splicing is an example of post-translational modification. Pay attention to the wording here. Yep. This is false. It's an example of post-transcriptional modification, not translational. This is with like, creates like that diversity within RNA transcripts. That's what that is. Okay. What happens when there is a high concentration of tryptophan? Yep, good job. The repressor will bind to the operator with high levels of tryptophan so that it blocks transcription from occurring. This means the RNA polymerase can't bind to the promoter and transcription won't occur. The reason it isn't the red one is because if we're already starting with high levels of tryptophan, transcription won't ever begin. But if the question was like, what happens if we start with low levels and then get to high levels, then that would be the correct answer because transcription would begin, but then terminate as levels get higher. All right, true or false? siRNA mediated risk cleavage of mRNA is a catalytic mechanism.
This is what Laura went over in class today.
Woohoo! Good job, guys.
Nice. Yeah. So siRNA kind of, or like RNAi in general, can like work as like a catalytic mechanism, meaning that they can like one siRNA can suppress multiple gene or mRNA. So it can affect a little can affect a lot.
All right, this is another one of those. A polypeptide with a positive charge at a physiological pH likely contains amino acids with R groups of which type? This is more of like the type of question she would ask you about this type of topic. So it's positive charge at physiological pH. Yeah, good job. So yeah, basic amino acids will have that positive charge. So associate positive charges with basic amino acids, associate negative charges with acidic amino acids, and then everything else is neutral. You don't have to know like this what specific amino acids are which. The two acidic ones are kind of easy because they have acid in the name, like glutamic and aspartic acid. But you'll have charts available if she does ask you a question like this. But this is more of like what you would see on a question like, what properties of the R groups can you infer from this type of environment? But you're likely not going to see too much about the chemistry of amino acids, but you could. So hydrogen bonding between separate subunits of DNA polymerase is an example of which level of protein structure? This one's a little tricky. Yeah, so this is why it's tricky, because the hydrogen bonding, you immediately would think secondary, right? But because of the fact that it says separate subunits of DNA polymerase, if you have more than one subunit, you have quaternary structure. So this is quaternary structure because remember, hydrogen bonding does occur in tertiary and quaternary. It's just the main form of bonding in secondary. So that's why like you should associate hydrogen bonding with secondary structure. But they still do exist in tertiary and quaternary. But because that keyword of separate subunits, that's why it's quaternary structure in this case and not secondary, because we have more than one subunit. That one's kind of tricky. I got it from Khan Academy. So if anyone has any questions, obviously you can say it in the chat, you can pipe up, whatever, but let's write it up. Okay, true or false? The signal sequence is cleaved after entering the nucleus.
Can proteins enter the nucleus and exit and then enter back in? It's a good question to think about answering this question.
Can I change my answer?
Sure. I don't know how you could do it on the thing, but
Actually, I can't.
unfortunately, but
Yeah, so signal sequence would not be cleaved after entering the nucleus because a lot of proteins enter the nucleus. For example, importin, it like enters the nucleus, it exits, and then it goes back in. So you wouldn't want to be cleaved, or like other proteins that like cycle in and out.
all right. What type of mRNA can code for multiple proteins? Monocistronic, polycistronic, siRNA, or miRNA? This is just like one of those terms that you should know because she might mention it with like no background and you might be like, I don't know what this means. Yeah, polycistronic. Polycistronic mRNA means that one singular mRNA molecule can code for multiple proteins. Eukaryotes do not have this ability, prokaryotes do. So prokaryotes can make an mRNA and have that code for a bunch of different proteins, whereas with eukaryotes, we have what monocistronic mRNA is. We can only make one protein per mRNA.
Tripalparon is an example of a polycistronic gene.
Yep. Where will a protein with no localization sequence end up? Yeah, good job. So proteins are made in the cytoplasm, right? They're made from the ribosomes, which are located in the cytoplasm. So proteins with no localization sequence are just going to stay there because that's where they're made. There's nothing to tell them where to go. There's no sequence that says you need to do this. The ER in the nucleus, I think most of you got with, you need that ER sequence or the NLS for the nucleus. With the outside of the cell, remember that if a protein needs to leave the cell, it has to go through the endomembrane system, which means that it needs that ER localization sequence to do that, which is why that is not the correct answer for this question. Good job. True or false? mRNA has to be transported from the nucleus to the cytoplasm before translation can occur. Yep, this is true. I guess I should have specified that this is eukaryotes, but I guess I just figured if you mentioned that there is a nucleus, you kind of would know that it's eukaryotes. But yeah, so eukaryotes, the mRNA has to be moved from the nucleus to the cytoplasm before translation can occur, because transcription occurs in the nucleus, translation occurs in the cytoplasm. mRNA in eukaryotes also has to go through that post-transcriptional modifications with like the 5' cap, poly A tail, splicing, all that stuff. Prokaryotes, the mRNA doesn't have to be transported anywhere because there is no nucleus. So the mRNA can just be transcribed and then translated like immediately after, or kind of, they happen at the same time actually, but they both happen in the cytoplasm because prokaryotes don't have nucleus. But yeah, for eukaryotes, this is true. A protein with one polypeptide can contain multiple domains. Did we already do this question?
Yeah, it's kind of similar to what we did earlier.
Oh, yeah. Okay. Yeah, this is true. One polypeptide can have multiple domains because those are just like different sections of the polypeptide. Best way I can think of to explain it. It can have subunits because more than one subunit, because one polypeptide is one subunit. But yeah, this is true. Prokaryotes have introns and exons. True or false?
The chart that we have on the slides we're going to send out will be helpful for questions like this too.
Jason, is Emily with you?
Yes.
Hi, Emily.
Hey, what's up?
If you were in Amherst, we would have come drop off stuff for you,
A
but
gift. Yeah, I
yeah.
know, but I'm not. I'm home,
Next time.
unfortunately. Yeah, seriously. Yeah, they do not have introns and exons. Only eukaryotes do. Prokaryotes do not participate in splicing because they don't have introns and exons, but eukaryotes do. So it's just one of those things you gotta know that prokaryotes don't have these. All right. What is the name of the general transcription factor in prokaryotes? I answered this earlier.
Good job. I think it was Ethan
Woohoo.
for asking about this.
Yeah. I should have put more fun answers. I couldn't think of any. Yeah, sigma factor. I don't think there is anything. I don't think there's any such thing as a beta factor or a regulatory factor. I kind of just made those up. There might be. I have no idea though. But yeah, the general transcription factor in prokaryotes is the sigma factor. The basal transcription factor, which is this red one, is the transcription factor for eukaryotes. But we talk a lot about sigma for prokaryotes. 5 more questions. The trip operon is an example of a blank operon, repressible or inducible. Yep, it's repressible. The lac operon's inducible. I just mentioned that, but you don't need to know what the lac operon is for this class. Repressible means that the operon's always on, but will be turned off in the presence of a molecule, in this case, tryptophan. That's what repressible means. All right, in low levels of tryptophan, the trip operon is off. True or false? Yep, good job. In low levels of tryptophan, the operon is on because of what I mentioned before. The trip operon wants to make more tryptophan. So when we don't have any tryptophan, we'd want the operon to be on so that we could make more. And NLS is a stretch of positively charged amino acids.
It's one of the only parts of like the signal sequence like chart that you actually like kind of need to know.
Yeah. Yep, that's true.
Good job.
Ooh. Eukaryotes can make more than one protein from one mRNA molecule. Also mentioned this before. Yeah, that's false. Eukaryotes can only make one protein from one mRNA molecule. We cannot make more than one. We are monocistronic, have monocistronic mRNA. Prokaryotes can, they're polycistronic. Important, needs to be bound to RAN-GEF to enter the nucleus. True or false? Yep, that's false. Important to enter the nucleus, it just needs to be bound to the cargo protein that it's bringing in. To leave the nucleus, it needs to be bound to RAN-GEF, but not to enter.
Just means bound to RAN, not necessarily GEF.
Yeah, not GEF, sorry. It needs to be bound to RAN, but not GEF. Don't listen to that, just RAN. Last one, GTPases can hydrolyze GTP and ATP.
Kind of getting at what we were. Oh, never mind. Sorry.
Yeah, this is false. This goes into the fact that enzymes are very specific. So GTPases can only hydrolyze GTP and ATPases can only hydrolyze ATP. It's never both. So that's just how they're very specific. That's it. Let's see.
Good job,
I know
everyone.
these aren't accurate because some of them are my fault. I'm sorry. So sorry about it. Christine, winners, yay.
Congrats,
all get-
guys. Good job, everyone.
Get high fives.
Does anyone have any questions about anything that we covered, but we didn't cover? If not, we can talk about general structure of the exam real quick, and then we'll be all set.
Also, this was the first time we've ever tried these like Kahoot reviews. We just wanted to see if it people like actually liked it. I mean, we can obviously go back to normal exam reviews like with the presentations, but we just figured it'd be a little more interactive. But so did you guys like this or do you prefer the original, whatever? You like the Kahoot?
I would say when I saw the Kahoot, that's when I tried to go to the meeting because if it wasn't for the Kahoot, I wouldn't have come, to be honest.
Right.
It sounds more fun than a regular exam review, doesn't it?
Because I feel like the thing I was thinking is we're kind of just reiterating everything that you've already like watched like with her lectures and like gone to her classes. So I feel like practice questions help a little more.
And we already had an exam review recorded for the same thing, so it doesn't make sense to do it twice.
Yeah, so we'll send out that recording, or Jason already did, but yeah, so we just wanted to see if people actually like this, but it seems like people did, so that's good. But yeah, so I'm glad you guys enjoyed it. I hope it was helpful. But yeah, if you have any questions, feel free to ask. You can email us, you can ask us now, up to you. But if not,
so
oh yeah.
Real quick, so just like general exam stuff. So I would basically just try to like read the question, pick out the important details. Laura often puts like extra information that's not important for the answer to the question, but
Oh yeah.
for like background information. So just try to figure out what the question is actually asking and answer every part of the question. Include as much details as like what she said is like pretend you're explaining to someone who's like not in the class, and like pretend you're not writing to the TAs, pretend you're writing to someone that's not in the class,
Mhm.
and include every detail that'd be necessary for them to like understand what you're trying to say.
Right, because it's easy to lose points or not get full credit if you like don't mention something, because if you don't mention a crucial part of like the answer, we're just gonna assume you didn't know it. We try not to be too, at least I try not to be too like harsh with grading. If I can understand what you're trying to say, and like I feel like you're not like giving me just like crap, like you should be fine. But with that being said, there's multiple TAs grading, and Laura also does grade. So right, like you're telling someone who, explain it like you're trying to teach a class, basically. Like you don't want to leave things out that are important.
Exactly. Also, like Laura mentioned in class, if you try to put like a lot of detail, but if you put some like extra detail that is wrong, that could like let you lose points. So just so you're aware of that. I try not to do that, but it's possible.
Yeah. So if you say something that's just like completely wrong, like if you're like a protein can't enter the nucleus fully folded or something, you'll like get points off because that's wrong. The exam is only all short answer questions. It's usually multiple parts. Yeah, I know.
The good thing is you have 36 hours to do it. It's all open book. So you don't have to worry about like memorizing things. You can figure it out, take your time.
I wouldn't suggest Google though, because odds you find the answers on Google are pretty slim, and it might confuse you more. I would like stick with your notes, stick with like the lectures, stick with all the resources Laura gives you. How many questions? There's usually
around
like 9 or 10.
8 to 10, yeah.
But they're multi-part, like they'll usually be like ABC. Some of them are like pretty short, like she'll give you like a list, like before she had like which one of these are like enzymes. And it's just like matching like the enzymes, like or whatever. Like some of it's like matching or just like not matching, but like filling in like really, really short, like it's just a one-word answer. Other ones are much longer. It depends on how much you want to write too. I mean, some people give us like essays, some people give us a paragraph.
Try not to give an essay.
Please try not to give an essay. If you feel like the only way for you to convey like everything you know or to get the answer right, like don't hold back, because we said don't write essays. Like obviously write as much as you feel comfortable writing. But I'm just saying, like some people write essays and some people write paragraphs and get the same amount of points. So
a lot of times I'll write a comment. I'll be like, you could have like just wrote this and got full credit. So
right, like I'll usually highlight a section and be like, this was like all you needed, just like for future reference. You won't lose points for being long. Laura actually likes the long ones. But yeah, so the only way you'll lose points is if you don't mention something that's crucial to the answer, or you'll mention something that's just wrong, like that's just not correct.
The closest thing to the exam questions are going to be the problem sets.
Yeah.
So I would definitely review those. That's like the type of questions that you're going to see on the exam. And like that's the kind of answers that like she's expecting. So like the feedback you've been getting from that, use that for like writing your exam questions. And also, just because you have 36 hours doesn't mean it should take 36 hours. Like it shouldn't take more than a few hours, honestly. If you're like prepared going in. So don't like stress about it too much.
Yeah, don't use Google, honestly, because you'll try to look something up and it just won't like it. Yeah, don't do Google. But use any of the resources she gives you, any of the resources we give you. You can email us during it too, but we'll probably forward it to Laura and she'll probably answer you, because all the TAs, we have very different schedules. But if you do have questions, email Laura. She will get back to you with like pretty quickly. She told me last semester, she was like, I literally was on my laptop like all literally every second of the day, like when the kids are taking their exam. So don't hesitate reaching out to her.
And yeah, so I'm about to send you this PowerPoint. It's got like 60 slides of like a lot of review stuff, so you can use that as a resource too as you're taking the exam.
Yep.
So
and it has nice pictures, like
nice
dogs, puppies at the end.
Yeah, it's got some puppy jokes in there. It's got some memes. So
Got some cats.
very
I think
interesting.
your cats are in there, aren't they?
Yep, my cats are in there too. It's not updated, so it doesn't have Rachel's dog, but
It doesn't have my dog. I just got him a couple left in January. That does look like him though.
yeah,
But
it's him.
that's not him,
This is
but-
a separate black lab.
Yeah, but they do look the same. But yeah, so if you guys have any questions, feel free to stick around and ask, or you can email us if it comes up later. But other than that, that's all we have for you. So if you don't have any questions, have a great night and good luck studying.
Thanks for coming, guys.
Yeah,
Thank you.
thanks.
Thanks, guys.
I have a quick question, sorry.
Sure.
Yep.
So for the trip operon, just to clarify, is it like it codes for proteins that synthesize or that that synthesize tryptophan? So it's like tryptophan is not like it's just like made within our body, right? Like it has to be taken from our diet and stuff like that, right?
It's a good question, actually. I literally should know this. I took the MCAT less than a year ago. I think we do get it just from our diet. Yeah, you're right. Yeah, so we just get it from our diet. Prokaryotes make it though. So when we talk about like the trip operon, like it's just in like bacteria that we're talking about.
Oh, okay.
Mhm.
All right. So it's just her that's going to be like, I mean, you said that you guys are going to be available, but her mainly 24/7 during the exam, right?
Yeah,
Yeah.
like we'll
Or-
probably be available, but I can't guarantee
Right.
you'll get a prompt response from the TAs because we all like, I don't know what all their schedules are.
Laura's the one that should be answering exam questions anyway, so
Yeah.
she knows like how much detail to give and everything. So definitely direct your questions to her directly.
Yeah, because like she could have a way better answer because like we don't know exactly what she's always looking for. But she would know. So yeah.
All right. Okay, that's it then. Thank you.
Yeah, no problem. Have a good one.
You too.